Welcome guys, this is Smriti from Sanu's Biology. Today I am discuss about this diagnosis of coronavirus. So, a person who get infected with this coronavirus, they have some symptoms like fever, cough, and difficulties in breathing and many moods. So, to confirm that they have this coronavirus in their body, they need to be tested. So for testing this coronavirus, there are two type of tests present. First one is RT-PCR test and second one is blood test. So firstly, we have to collect sample for any kind of test. For uh, for RT-PCR test, we collect the sample with the help of swab, and this collection called nasopharyngeal swab collection. So we collect the sample with the help of this swab. We insert the swab in this direction from nose and collect the sample. And this swab containing many droplets, and in these droplets there are present of many viruses. So we extract the mRNA from those viruses and we start the procedure of RT-PCR. So for RT-PCR we need some primer. These primer are short nucleotide, and there are present of different primer. So reverse transcription PCR have different principle, and the principle of this PCR is that there is present of some small short nucleotide primer which is known, and this help to extend the mRNA, and from this we get the cDNA. So firstly, this primer is made from the known sequence of coronavirus. So as we know the working principle of PCR, the double-stranded DNA or RNA get separated, and this primer bind at the starting point and DNA polymerase make them extended. But here we use mRNA, so here reverse transcriptase enzyme help to get our cDNA. Now this cDNA amplify into DNA and we get lots of DNA. Now we perform this reverse transcriptase in this type of PCR machine, and then we go to real time PCR. In this we take help of cyber green, which help us to get the result for positive coronavirus. So as we know that this cyber green get glowing after binding with the DNA molecules. So this cyber green generally bound to the space of minor groove of DNA segment. When this DNA segments get separated, there is no minor groove. So this blue color of cyber green here in diagram, it is not bounded with the with the strand of DNA. So when this DNA get double stranded and they get amplified, so there are bounding of this cyber green, and this bounding of cyber green makes glow, and as we observe the result, there is fluorescence. As the PCR cycle going forward, there is increasing in fluorescence, and so we detect that there is present of coronavirus. If there is negative result, then this primer doesn't get bind with this DNA, and there is no fluorescence and there is no any kind of graph. So we conclude that if there is no fluorescence, it is negative corona, and if there is fluorescence, it's so positive result for corona. Now we come to the blood test. This is second kind of test, and this is uh, just started in few days. So this kind of test have immunoglobulin, and we work with immunoglobulin in this test. So as we know, when anyone get infected, there is primary antibodies and secondary antibodies also release. So this is blood test for COVID-19. Here uh, the here present of IgG, IgM, SB indicate that this B1 is for the buffer, for this buffer slot. This S is used for our sample, and IgM, IgG uh, is for visualizing the result. There is an important reaction behind this between antibodies and antigen, as we know in immune system, and this C is for control. So how does this test is performed? Firstly, we need to collect our blood sample. There is no need to collect lots of blood, so we use only we used to prick our finger and collect only 10 microliter of blood, and put into S slot. This is for sample, and here we put some drop of blood, and then for we wait for 10 to 15 minutes. As this buffer run, we got result. So for result prediction, we saw that if there is a line of C in upper part, it shows that your test is going correctly. So and this also indicates that your kit is perfectly work. And there is IgG and IgM. If there is line in IgG, IgM, or in both, it's so positive. And if there is no line in both IgG and IgM, it shows negative. And the principle behind this is about immune system, that if anyone get infected with coronavirus, they release this antibodies, which is IgM firstly. And if it is a if it is very long period of infection, then it also comes in IgG. If newly infection is only there, it's come in IgM. If it is very old, if it is very longer time, then it also come into IgG. if if you have if you have infected more times, then it comes only in IgG. So these three types of results so positive, and only if there is a C get line, then it is negative result. And if there is this type of showing any result, it is invalid. Your test kit is not working properly. And then what is result? This is negative result. See here, there is only lining in C portion, and here is both this T generally for test, and it have IgM and IgG antibodies here. So thank you. Please like, share and subscribe my channel.